Okay, let's go ahead and get started. So we left off last week just beginning our discussion of metabolism, which is everybody's favorite topic, including mine. I hope you recognize my sarcasm. So one of the things that I think is underappreciated as we talk about metabolism is everyone realizes that one of the main things that come out of metabolism is energy. And they forget that there are other commodities, there are other reasons why understanding metabolism is very important. One is that another, and actually you can kind of group this if you want to with energy, is the production of reducing power. So in this particular case, obviously what I mean is electrons. So here in metabolism, so as we talk about glycolysis first, you're going to see during oxidation steps the concomitant generation of NADH, a reduced electron carrier. Those electrons can and will be moved to something else, hopefully to produce energy, but they can also be utilized, for instance, to reduce iron, as iron is brought into the cytoplasm of the cell, free it from a siderophore. So reducing power, which is generated here in these oxidation steps. So you can always recognize an oxidation step in any of these metabolic pathways because you will always see concomitantly the production of either NADH, FADH2, or in some cases NADPH. So some form of an electron carrier is produced, and those electrons can be used to do work in other places. And then lastly, another commodity that's underappreciated that's generated by metabolism are precursor molecules. So you've all learned the TCA cycle as a cycle. You start off with acetyl-CoA, going into citrate, and you think of the carbon skeleton going through this as going all the way through. But in reality, there are various siphons within the cell that exist that are pulling off molecules, for instance, at alpha-ketoglutarate. So alpha-ketoglutarate can go on to succinyl-CoA, but it can also be pulled off to form amino acids in anabolic pathways. The same thing with succinyl-CoA. It can continue on the TCA cycle, but it's also an important precursor for porphyrin rings. So the cytochromes that have prosthetic groups associated with them require this. So virtually all of these metabolic pathways never go 100% to completion because somebody is always pulling something away from this for another purpose. So just remembering that there are other commodities besides energy in the form of ATP are being produced in this. Okay, so now we can't avoid it any longer. Let's go to glycolysis. And remember, I am not a biochemist. I play one here in front of class for about 2 lectures every year. I don't expect you to become biochemists in this class, even though this class counts towards your biochemistry minor, which is great. I don't expect you to memorize this pathway. As a matter of fact, I ask that you don't memorize this pathway. As we talk about metabolism, we're going to talk first about glycolysis, then we're going to talk about the pentose phosphate pathway, we're going to talk about the Embden-Meyerhoff pathway, we're going to talk about, what else, the TCA cycle and electron transport chain. There's a lot of places in there where you will be tempted to memorize and probably have already memorized them. What I want you to do in this class is to view these various metabolic pathways from 10,000 feet and to see how they fit together, to appreciate how these things work. So my philosophy is kind of this, is that, like when I was in college and in graduate school, when I took biochemistry classes, I kind of lost the appreciation for how metabolism worked because I was so concerned with memorizing what enzyme happened where and what was the order of the steps that I never felt like I truly appreciated metabolism for the significance that it has. So what I'm hoping is that those of you who are either taking biochemistry now, have already taken it, or will take it with somebody like Lisa Landino or Doug Young, or Randy Coleman, who are biochemists. This will actually help you complete your understanding of metabolism. So we're going to talk about some of the individual parts of it, but in reality, I don't want you to memorize glucose 6-phosphate goes to fructose 6-phosphate. Some of the things that I want you to recognize, though, are the fact that glycolysis exists in two different stages. So the first stage above the line here has no energy given off from it. As a matter of fact, energy is put into this. So you can see that ATP phosphorylates glucose, and ATP in this other step phosphorylates fructose. So at this point, energy is actually being put into the system rather than coming out of the system. So no energy yield, all energy input at this point. Now, most hexoses, or 6-carbon sugars, are converted, so bacteria in the environment, unless they're living in association with us, very seldom have access to very much glucose. Glucose is relatively rare in the environment, but other sugars are present. So we've talked about mannitol, we've talked about mannose a little bit, at least in names. All of those other sugars that bacteria can use at some point get converted to something that fits into the glycolytic pathway, generally at the point of fructose 1,6-bisphosphate. So pathways that break down these other sugars are less efficient than glucose because they have to have several enzymatic steps to bring them to a molecule that will fit into this glycolytic or Embden-Meyerhof-Parnas pathway. That's why, as I mentioned earlier, when we talked about group translocation, E. coli will always, every time without fail, suck glucose out of the environment, even until it's absolutely exhausted, even if that environment is rich with other carbon and energy sources that it can use, because glucose will fit directly into this pathway. Other sugars have to be enzymatically processed in order to fit something in. So glucose is the most efficient carbon and energy source for any bacterium, but it's a relatively rare molecule in nature, except microbes that exist in in association with the vertebrate host. So that 6-carbon stage is entirely preparatory for the second stage, which is below that line. In the second stage, this is where you're going to begin to see energy. And I'm sure you all remember this, but I'll remind you that the glycolytic pathway, although it does produce energy, produces a very modest amount of energy. If a cell has to rely entirely on glycolysis to generate ATP, it has to be exceptionally good at glycolysis. And there are bacteria that rely exclusively on the glycolytic pathway to produce energy. You know, you and I can use glycolysis, but then we can feed into the TCA cycle, the electron transport chain. Bacteria like, for instance, Treponema pallidum, the spirochete that I showed you the video of it, little corkscrew. That bacterium doesn't have any other pathway for energy production other than glycolysis, but it just does glycolysis so well. The streptococci that live in our mouths right now that are potentially, hopefully not, but potentially contributing to tooth decay, those are organisms that have to rely entirely on the glycolytic pathway. But they're very, very good at it. They're much more efficient glycolytically than we are. Because the glycolytic pathway inherently does not produce a large amount of ATP. So this second stage, which begins here, is where that 6-carbon molecule, in this case fructose 1,6-bisphosphate, is split into glyceraldehyde 3-phosphate and dihydroxyacetone. Short-lived. In essence, you could just view that as 2 glyceraldehydes. Now, this is what I want you to recognize. I want you to be able to recognize that there is one oxidation step in this pathway. You can always recognize it because, remember, by definition, an oxidation is taking an electron from something. And electrons never exist free in nature. They have to be associated with a carrier. So here, we have a carrier, nicotinamide adenine dinucleotide, or NAD+. So when you see NAD+, this is the oxidized form. When it becomes reduced, it takes on an electron. Actually, it takes on a pair of electrons. And then it also takes on proton to help balance that. So you can always recognize the reduced form of these electron carriers because they have an H after them. The most proper way to express this is NADH plus H+. And H plus doesn't actually exist free in the environment. It exists as a hydronium ion, but I'm going to leave that to your Gen Chem professors. But in any case, this is the sole oxidation step in here, and you can recognize it. But it sets up the two other things that I want you, or the two other steps that I want you to recognize the significance of. I don't want you to recognize the substrate or the product, but I want you to recognize that here is an example of a substrate level phosphorylation. So what is happening at this step is ADP is being turned into ATP. So there is a high energy phosphoanhydride bond, for you chemistry people out there, being generated here. That phosphate group is coming off of the substrate, hence the term substrate level phosphorylation. It's important, if for no other reason than for the purposes of the exam, that you can recognize and know the difference between substrate level phosphorylation and oxidative phosphorylation, which we'll talk about later. That's as the result of electron transport chain. So here you have a step where you have substrate level phosphorylation. There's one more step at the end where you're going from this high energy phosphoenolpyruvate. Remember, we talked about this molecule earlier too when we talked about group translocation, because PEP is actually used as the energy source for group translocation. So this high energy phosphate bond from PEP can feed group translocation to bring in glucose, for instance, or it can participate in a substrate level phosphorylation where ADP is turned into ATP, and then pyruvate at the end. So I don't know how biochemists define the beginning and the end of glycolysis, but I define the end of glycolysis as the production of pyruvate. So if you look at this overall, you can see again the modest amount of energy that's produced here. Only 2 ATP molecules, and pyruvate, which is kind of the end product here, is actually still a very energy-rich molecule. It's still highly reduced. It could still produce a lot of energy, and you know, and I know how that happens, because you and I know that this can go through another step and go into the TCA cycle, and we're going to get to that. But first, I want to talk about the pentose phosphate pathway because it relates to the glycolytic pathway. Another term for the pentose phosphate pathway is a hexose monophosphate shunt. The term shunt is important, because what can happen in the pentose phosphate pathway is that glucose 6-phosphate, which is one of the early, actually after the first reaction of glycolysis, can be shunted into this pathway. Now, we consider the pentose phosphate pathway part of catabolism only because it's producing a lot of different molecules, such as ribose, which is a 5-carbon molecule, acetoheptulose, which has 7 carbons, a 3-carbon molecule, 5, 6. So all kinds of carbon skeletons are being produced in the pentose phosphate pathway. So hence it is a catabolic pathway. The really most important thing about the pentose phosphate pathway is that it's crucial for producing precursor molecules. So thinking just about, for instance, xylose 5-phosphate. This is a 5-carbon molecule. You can pretty much guess what that's going to be used for, purines and pyrimidine synthesis. So again, thinking about siphons, taking stuff off of catabolic pathways. What the pentose phosphate pathway is doing is that it's playing a critical role in anabolism, the building of molecules. So all of these various carbon skeletons that are produced, and the key enzymes here are transketolase and transaldolase. But these carbon molecules or carbon skeletons are being used for amino acid synthesis, for lipid synthesis, for nucleic acid synthesis. This is what's going to happen when a cell is under energy replete conditions. So I'm off on my bullets here a little bit. But this pentose phosphate pathway is what a cell can do when it has lots of ATP. If a cell doesn't need much ATP, because glycolytic pathway has worked very well, it needs now to use some of that energy to grow, to build to reproduce, to produce more proteins, more amino acids. And so molecule here, glucose 6-phosphate can be shunted from the glycolytic pathway into the pentose phosphate pathway to create all of these important molecules in the biosynthetic pathway. So these enzymes, especially transketolase, generate these 3 to 7 carbon sugar phosphates, which are really critical for various biosynthetic pathways, such as the nucleic acid synthesis. Here's just another way to look at the pentose phosphate pathway. I kind of like this one because you'll note that the pathway here yields no usable energy. No place in this pathway do you see ATP generated, GTP, or NADH. Parts of this pathway can, when the biosynthetic pathways are completely saturated and everybody is happy and they don't need to grow anymore, these molecules can be shunted back into the glycolytic pathway. So this pathway can generate energy, but not directly. It can pass fructose 6-phosphate or glyceraldehyde 3-phosphate back into the glycolytic pathway to continue substrate-level phosphorylation. But the pathway itself, the pentose phosphate pathway, does not produce any usable energy, not even in the form of NADH. And again, here are some of the precursor molecules for aromatic amino acids from erythrose. So this is very active when energy levels are very high in the cell. The other thing, I'm going to stay with this slide for a minute, is you'll notice that there are oxidation steps in here, and you can recognize them very easily. Here's an oxidation step, nicotinamide adenine dinucleotide phosphate. So this is a different species of electron carrier. It differs from NAD only by the addition of a phosphate group, but you can see that it becomes reduced to NADPH. Here's another oxidation step with the generation of NADPH, and you notice that these are multiples. So this pathway produces a large amount of reducing power. Again, one of the commodities that catabolic pathways create is reducing power. But let's differentiate this species of reducing power, NADPH, from NADH, which is produced by the glycolytic pathway. The NADPH that's generated by the pentose phosphate pathway is critical for anabolic pathways. Many of the pathways that are used to synthesize amino acids, nucleic acids, lipids, require at some point during that biosynthesis reduction. They require a source of electrons. So these electrons on NADPH are not going to be shuttled into the electron transport chain later to do oxidative phosphorylation. Instead, they're going to be, think of them as nails in a carpenter's toolkit. These are molecules that are going to donate their electrons into the biosynthetic pathways rather than into energy-producing pathways. But it also it reinforces the point that I'm making ad nauseum, that one of the critical commodities of catabolism is the generation of precursor molecules. And in this case, we're talking about an energy source for biosynthesis. So here's yet another way to look at this, and this is kind of an old slide. It doesn't project very well, and I like it only because you can again see these oxidation steps quite clearly and all of the different carbon skeletons that be produced. But you can also see how the fact that molecules from this pathway, if they are not siphoned off for use in a biosynthetic pathway, can simply be shunted back into the glycolytic pathway to produce energy. So it's a siphon that's coming off of that pathway depending on what's needed by the cell at that moment. This would be active under energy replete conditions, and if energy is needed, then molecules would tend to stay in the glycolytic pathway. Now, one of the cool things about the glycolytic pathway that you've all learned, the Embden-Meyerhoff-Parnas pathway, is that it's the same in your cells, it's the same in your pets' cells, it's the same in virtually every cell of everything that you know and love. But bacteria actually have alternative glycolytic pathways. And I'm only going to talk about one, because I only know anything about one of the other ones. And this is actually perhaps the most original glycolytic pathway. So this evolved in bacteria, and it still exists to this day only in bacteria. It's called the Entner-Duerdorf pathway. It's actually highly related to the pathway, and so I don't want you to know anything about the pathway here, at least in terms of the molecules. The end product, again, is pyruvate, just like glycolysis. But this is a really interesting and important pathway. Bacteria, this is a pathway that's very important in our intestinal tracts right now. So part of the way that we maintain a mutualistic interaction with the microbes in our intestinal tract is that we are literally feeding them, not simply with the food that we eat, but also with the mucus that we produce to protect our intestinal tracts. Bacteria in our intestinal tracts actually stimulate cells in the intestinal tract to produce mucus. One of the things that mucus is rich in is gluconic acid. Gluconic acid in these bacteria feeds into the Entner-Duodorf pathway, and they use that as their glycolytic pathway. So it's a really important pathway to understand in terms of humans. But it's also very important from an economic reason. So this is the one that everyone in every textbook always cites because you're trying to always keep undergraduates' attention. And what keeps attention better than probably anything except sex is alcohol. So there is a bacterial genus and species called Zymomonas mobilis, which is a bacterium that lives in association with the agave plants in Mexico. It occurs on the leaves and I think to some extent on the roots. I'm not sure whether it's leaves or roots. I'm not a plant person. But when people make pulque or cactus beer, the fermentation that's done traditionally is done not by yeast, as most of you think of alcohol as being produced by yeast, which is a eukaryote, alcoholic fermentation. Instead, traditionally, cactus beer or pulque is made by fermentation by a bacterium, Zymomonas mobilis, which uses this unusual pathway to produce ethanol. And this cactus beer or pulque actually ends up being distilled into one of my favorite cocktails, is tequila. And now, I don't know, honestly, whether the unique flavor or the unique effects of tequila have anything to do with the fact that it's coming from cactus or whether it's coming from this bacterium. But I like to think that Zymomonas mobilis and this Entner-Duodorf pathway producing alcohol is actually plays a role. In reality, I'd like to think that now most modern distilleries don't use the traditional methods of fermentation anymore, so they actually use yeast. But traditionally, this is the organism that's responsible for the production of tequila. And I think still in some perhaps small distilleries it may still be. Okay, so again, just to show you a little bit about the diversity. So remember, one of my major objectives in this class is to teach you that metabolic diversity and genetic diversity really all lie within the prokaryotes. Here are the three different glycolytic pathways. You and I have only one. We have the Embden-Meyerhof pathway. But other bacteria have the Entner-Duodorf pathway. We're not going to talk a bit about the phosphatidylase or heterolactic pathway because I don't know a damn thing about it. But you can also see that some bacteria actually can use both. They can switch back and forth. And some of the bacteria that use the Entner-Duodorf pathway are some pretty serious pathogens, like the bacterium that causes cholera. Pseudomonas aeruginosa, which is a major opportunistic pathogen in cystic fibrosis patients, in burn patients, and in cancer patients, also uses exclusively the Entner-Duodorf pathway. Agrobacterium tumefaciens, which we'll talk about later, which is the underpinning of all transgenics in plants, uses the Entner-Duodorf pathway. So these are not minor little exceptions to the rule on glycolysis. These are majorly important ecological organisms. Now, at the end of the glycolytic pathway, or at least as I define the glycolytic pathway, is pyruvate. You and I all love the products of fermentation. Even if you're a teetotaler and you don't like alcohol and beer and wine, things like cheeses, sauerkraut, fermented food, all depend on bacterial fermentation in order to be created. So for us, it's beer and wine and Swiss cheese, but what does that do for the poor microbe? Why is fermentation so widespread in bacteria? It's largely because of the fact that this oxidation step, remember there's one oxidation step in the glycolytic pathway, recognized again as NAD in the oxidized form to NADH plus H. This electron carrier is actually in a fairly small supply within a cell. This electron carrier, because there are so few molecules of it, runs the risk of being entirely reduced if it's not regenerated. If all of the NAD plus is reduced to NADH, oxidation stops. The glycolytic pathway stops. There's no place for the electrons to go. It's like a clog in the drain of your sink. Water can't go down it any longer. It's the same thing here. So this is what fermentation does for bacteria. For the most part, fermentation generates no more energy for the bacteria. All it's really doing is serving as a place to dump that electron. This NADH molecule needs to get rid of the electron. So one of the things it can do, and this is the simplest form of fermentation, and it's a very widespread form, this is what streptococci and lactic acid bacteria do, is they simply dump the electron onto pyruvate, reducing pyruvate to lactate, or lactic acid. This is the acid that streptococci in our mouths are producing, that those of you who are going to dental school are going to profit from as people's cavities need to be filled. But there are other, so in this particular case, there is no benefit to this bacterium because this lactic acid will now simply be secreted as a waste product. The advantage to the bacterium is that it's regenerated its NAD pool so that it can continue to oxidize glyceraldehyde 3-phosphate. So it's a simple process, but the bacterium is still, in this case, wasting a very energy-rich molecule. Other fermentation pathways, other bacteria have evolved a whole host of different pathways of varying complexities. But in every case, the one thing that you will always see in any fermentation pathway is the oxidation of NADH to NAD. That is what a fermentation has to do for the bacteria. The end product, if it's useful to us, that's great. The bacterium doesn't really give a damn if it's giving us a useful product. It's just allowing its glycolysis to continue. Now, again, in terms of biodiversity, in terms of genetic and metabolic diversity, fermentation is a great example of how bacteria have evolved a great deal of diversity. So I've already kind of harped on the lactic acid bacteria like streptococci. That's what's shown here in number 1 on the top. This is the simplest form of fermentation. NADH is dumps its electron onto pyruvate to give lactic acid. But then you can look at, for instance, yeast, or our favorite bacterium that produces tequila, Xymomonas, which has a slightly more complicated fermentation pathway with an intermediate of acetaldehyde. But again, you see that NADH is oxidized back to NAD, and you get the useful for us product, ethanol, from that. But then look at all of these other products that are present here as fermentation end products. Acetic acid, propionic acid. Propionic acid is what gives Swiss cheese its very distinctive flavor. So you'll notice that this fermentation that's going on here, you can see is very elaborate. So from pyruvate it goes through oxaloate, malate, fumarate, succinate, finally propionic acid with the production of carbon dioxide. That carbon dioxide is significant too because that's the gas that causes the eyes in Swiss cheese. And then propionic acid gives it that flavor. I forgot to mention that bacteria that, like streptococci, that can only do this fermentation where pyruvate is turned into lactic acid, don't have TCA cycles. They don't have electron transport chains. And remembering that these microbes use catalytic pathways to generate precursor molecules, when you have a bacterium like strep that relies exclusively on fermentation rather than the electron transport chain and TCA cycle, it can't make a lot of its own precursors. So bacteria like strep have very difficult nutritional requirements. When we grow them in the laboratory, we have to baby them. We have to spoon feed them amino acids and nucleic acids because they can't make their own, because they don't have a TCA cycle, because they've evolved to live in high carbohydrate environments like our mouths, for instance, where they can just rely on glycolysis. Now, again, this is just another example of some of these various fermentation pathways that bacteria use. So E. coli is what's known as a mixed acid fermenter. This is going to be important for you in the laboratory because we're going to actually look at mixed acid fermentation. So during fermentation in E. coli, it not only produces ethanol, but lactic acid, acetic acid, formic acid. This is why you don't use E. coli to brew beer or wine, because while it is producing ethanol, it's also producing all these nasty tasting things. And who the hell would want to drink something that was fermented with a bacterium that normally lives in your colon? But other bacteria, like the Enterobacter, which we'll use in the laboratory, are what are known as a butanediol fermenter. So they produce all of these various end products. Our favorite, alcoholic fermentation, ethanol and CO2. And then you can have some really cool ones like Clostridium. Clostridium produces butyric acid, butanol, isopropanol, acetone, and carbon dioxide. But this is a really important fermentation for lots of different reasons. So again, if you look at fermentation in this way, so here's pyruvate at the center. So this is the starting point for every fermentation pathway. Here's the Clostridia group of bacteria that will take the fermentation through acetyl-CoA. Sometimes they'll stop at acetate, other times they'll go to butyrate, sometimes to butanol, sometimes to acetone. As it turns out, this is a very historically important pathway. In World War I, the Allies were running out of acetone. They used acetone to make gunpowder. So there was something referred to as the shell crisis during World War I, where they literally could not make enough shells. The way that the Allies were making acetone is they would take 100 tons of hardwood and take it through a process called pyrolysis. 100 tons of hardwood, which took years to grow, to make 1 ton of acetone. Pretty soon, England had pretty much cut down all of its hardwoods, and so was running out of acetone. So the Prime Minister turned to a biochemist slash microbiologist named Chaim Weizmann at Manchester University and asked him to come up with a better way. And so what Weizmann did was he used Clostridium acetobutylicum, now better known as the Weizmann organism. Chaim Weizmann, because he was a clever biochemist and a good microbiologist, realized that he could feed these bacteria, rather than hardwood, grains. So he could take 12 tons of grain, as opposed to 100 tons of hardwood, and produce 12 times as much acetone by allowing this bacterium to ferment and produce acetone. So this is really what solved the shell crisis. And almost 100 years ago today, it's 100 years ago in November, the Balfour the Balfour document was signed. And in that, what the Prime Minister of England did was, in appreciation for what Chaim Weizmann had done for the war effort, promised that England would do whatever it could to come up with a homeland for the Jewish people. Chaim Weizmann was one of the leading Zionists in England at that time. And this actually helped to create Israel years later. And even more important than that, Chaim Weizmann becomes the first president of Israel. So Israel's genesis is really due to this bacterium fermenting acetone. And it was all recognized by a biochemist, microbiologist, who now becomes president. So literally anyone can grow up to be president of whatever country they want to. And I'm going to skip this for a lack of time because I kind of tried to give you a nutshell of that. But here's the other cool thing about Clostridia fermentation. It's not just important in acetone fermentation. You and I are creating butyric acid by that same bacterium or groups of bacterium in the intestinal tract. We all know that a high fiber diet is beneficial to our health. We all know that a high fiber diet helps to protect you from colon cancer. Well, it's not simply that the fiber is doing anything, because fiber, by definition, is indigestible carbohydrates. These are things like cellulose, pectin, the things in the plant materials that we don't have the capabilities to digest. Bacteria like Clostridia and Bifidobacterium, or Bifidobacteria, shown here as these bifurcating bacteria, can actually ferment those to short-chain fatty acids like acetone, but also, very importantly, butyric acid. So this is a fermentation product of materials that are in our diet that we can't digest, but the bacteria can digest them very well, especially the Clostridium. Butyric acid, as it turns out, ends up being the major energy source for the cells that line our colon. We don't have to feed using glucose the cells that line our entire colon. Instead, what they're doing is they're taking butyric acid from the lumen and feeding that directly into the mitochondria of the colonocytes, where it undergoes beta oxidation and allows those cells to reproduce. Now, that's cool, but it also helps to suppress colon cancer. If a cell becomes cancerous, one of the things that happens in a cancerous cell of colon cancer is the physiology switches and glucose metabolism is ramped way up, and the mitochondria are largely kind of shut down. What happens is that butyric acid, if it's not being utilized in the mitochondria of that now cancerous cell, it has the ability to inhibit histone deacetylases inside of the cell. So that changes the gene expression within that cell to promote apoptosis. So as a cell develops cancerous, becomes cancerous as it undergoes that transformation, the butyric acid produced by the Clostridium in our intestinal tract has the possibility to induce apoptosis, to cause that cell to die before it actually turns into a tumor, which is really cool. And this butyrate has a lot of other effects. It actually, and probably outside of the realm of the discussion right now anyway, is that it affects the immune cells by stimulating T regulatory cells. It has a tremendous healthy effect. This is why a high fiber diet is important. It's not the bulk that it gives us. It's actually because we're feeding our microbiota in the intestinal tract. So understanding bacterial fermentation is very important. It's also important because it underpins many of our diagnostics. So for those of us who are interested in pathogenic microbes, one of the things that has to be done in the laboratory with clinical samples from people who are infected is the bacteria need to be identified. And one of the major ways that we use to identify microbes, and you'll do this in the laboratory, is to look at the fermentation products. So we can do what's known as a methyl red test. You'll do this test quite a few times before you finish this class. What we're doing here is looking for bacteria that do mixed acid fermentation, because a bacterium like E. coli that does mixed acid fermentation will have a low terminal pH. And so if we put a pH indicator in here, methyl red, which becomes actually unusually red if it's acid, and that's very much the opposite for most pH indicators. But that's an indication of that type of fermentation. So we can eliminate a lot of bacteria, such as bacteria like the Enterobacters, which we also deal with in the lab, which produce a neutral end product to fermentation, butanediol. So we can test using what's known as the Voges-Proskauer test for an intermediate in the fermentation pathway of butanediol. So we can find positives there, which will tell us that this bacterium is not an E. coli, it's not related to E. coli, instead it's possibly Klebsiella pneumoniae or Enterobacter species. So these fermentation pathways are very convenient biochemical assays for us to diagnose or to differentiate bacteria. And they'll be very important for you in the laboratory. Okay, end of the glycolytic pathway, thank God. No more fermentations. Let's talk about what happens. We could be talking about what happens in your cells and my cells, but now I want to talk about what happens in bacteria that, rather than simply rely on glycolysis, have this TCA, citric acid cycle, whatever you want to call it. So once we get past pyruvate and get into the get into the TCA pathway and then the electron transport chain, now what we're talking about is respiration as opposed to fermentation. So the early steps in that, and now what we're talking about respiration are glycolysis. So now we're beginning at the end of glycolysis. Pyruvate molecules are being fed into the citric acid cycle. CO2 is being removed. You can see here that there's an oxidation step. Now, again, remember, I don't want you, I'm asking you, I'm begging you, please don't memorize these pathways, at least not for this class. maybe for biochem class, for Randy or someone. But there's energy production here. But the energy that's produced by the TCA cycle is also very modest, at least in the terms that you think of energy as a high energy phosphate bond. At one point in here, there is a substrate level phosphorylation, but now it's not ADP that's being phosphorylated, it's guanosine diphosphate being substrate phosphorylated into GTP. Again, recognize that this is a substrate level phosphorylation because the phosphate that's being put on here is coming from the substrate being oxidized, that's being run through this TCA cycle. Now, this is where the TCA cycle really shines, these next 2 points. One is the production of precursor molecules. So the glycolytic pathway produces precursors, but not as many. You can recognize that there are not as many steps in the glycolytic pathway as there are in the TCA cycle. But virtually every one of these intermediates in this TCA cycle is a precursor for some type of a biosynthetic pathway. Aromatic amino acids, nucleic acids, fatty acids, all are sucking intermediates off of here. I'm not interested in you understanding that alpha-ketoglutarate is used in certain amino acid structure. That's not the important point. The important point is that one of the crucial things about this pathway is that it generates so many precursors. And here's the other place that the TCA cycle really shines. What really makes it so energy efficient is despite the fact that it's only doing one substrate level phosphorylation, it's producing a huge amount of reducing power. Again, coming back to those 3 commodities that catabolism always makes, reducing power. So you can see the oxidations here. Here's an oxidation, you can see the NAD is being reduced. Here's a second one, here's a third one, here's a fourth NAD, and there's yet another electron carrier, flavin adenine dinucleotide, which can be reduced. So because of all of these oxidation steps, there are a lot of electrons being taken off of these molecules. As we're going to see in the next part of this section on metabolism, these NADH molecules and even this FADH molecules, these reduced molecules, can now use those electrons, not for biosynthesis, like NADPH, but instead can put these electrons into the electron transport chain to drive the end of respiration, the oxidative phosphorylation, which is respiration. So here's kind of not a great slide, but it's for comparative purposes in terms of the production potentials of both of these pathways. You can see glycolysis on the left and the TCA cycle on the right. And so what you can see is substrate level phosphorylations. I put these little red and yellow hash bars. You can see 2 substrate level phosphorylations in glycolysis, but only one in the TCA cycle. So in that way, glycolysis wins that cycle. But look at the generation of reducing power. And remember that reducing power equates to energy down the line via the electron transport chain. There's only one oxidation step, so there's only one point in glycolysis to generate reducing power, as opposed to all of these sites, and the FADH is not even shown on this thing. So here you have much more reducing power. So this is the beauty of the TCA cycle. It's producing all of these electrons from all of these oxidations, which are eventually going to be used to generate energy in the electron transport chain. So now we're beginning to come to the end point. So here is where oxidative phosphorylation is going to become, or is going to come to be. So remember, one of the things that you need to be able to do is to contrast substrate level phosphorylation, so the generation of ATP by taking the phosphate group from the substrate, from oxidative phosphorylation, which is the generation of ATP at the end of the electron transport chain. So here, we've gotten to the point, so the electron transport chain, remember, invented by bacteria, co-opted by eukaryotic cells billions of years ago into what we now call mitochondria. The process is largely the same. Bacteria have more biodiversity in terms of the actual pathway of the electron transport chain, but they're still accomplishing the same thing that our mitochondria are. So here, in the cytoplasm of this bacterium, you have the reducing power, NADH, that either came from glycolysis, or the Krebs cycle, or the Ender-Duerdorf pathway. Just like fermentation, the cell needs to regenerate NAD. Otherwise, all of those oxidation steps stop. So if this step, the first step of the electron transport chain, did nothing else, it would still be doing the cell a big favor. It would still be allowing the cell to survive. Because the first step here is that the electrons from NADH are used to oxidize the first step in the electron transport chain, this flavin mononucleotide protein. And again, no memorization. I'm not interested in you knowing the order of proteins that the electrons go through. The important point is that NAD is regenerated. So the reduced form is oxidized, NAD is regenerated, the cell can breathe a huge sigh of relief, it can continue with glycolysis, it can continue with the TCA cycle, so it can produce energy. But unlike fermentation, where that electron just went on to a piece of junk that turned into a waste product like lactic acid or propionic acid, now the cells have evolved the ability to use those electrons not just as a waste product, but to pass them along this electron transport chain. So it passes it from this component to coenzyme Q, and then the electrons are passed on to cytochrome B, to cytochrome C, to cytochrome A. The point to notice here is that, remember that electrons are negatively charged. So at various points, as the electrons come onto another molecule in this electron transport chain, to balance the charge, these proteins, or sometimes they're not proteins, these electron transport chain intermediates, will take up protons from the cytoplasmic side to balance them. So here, when these electrons came from cytochrome Q to coenzyme Q rather, to cytochrome B, notice that there were protons that came across. In reality, what's going to happen to those protons is they're going to be extruded to the outside as the electrons are passed on. Because once these electrons leave cytochrome B, those two protons are now unstable. They're giving a positive charge to the protein. And the protein, rather than dump them back into the cytoplasm, this is the other important point, dumps them onto the periplasmic side, or the extracellular side. So now you're beginning to see how that proton motive force was built. We talked about proton motive force as an energy source to drive flagellar rotation. We've talked about the proton motive force as a way to drive active transport through symporters and antiporters. So here's where you're beginning to generate it. So as the electrons are going on and being passed along, these protons are being dumped to the outside. So in this particular case, you can see in this electron transport chain shown here, you have three what are referred to as coupling points, where the transport of protons is coupled to the transport of the electrons. So two protons are dumped out here, two protons are dumped out here, two protons are dumped out here. And all of those protons came from the cytoplasm. So you're building a battery. You now have a stronger positive charge on the outside than on the inside. And so now what can happen here is oxidative phosphorylation. That proton motive force can now come back through to the cytoplasm, reducing that proton motive force, reducing the potential energy. And that decreased potential energy will be captured in torque, in rotation, similar to the flagellum. We talked about the mechanism of how these protons came across through MOT A and MOT B to generate rotation of the flagellum. Now, when they come through this F0, F1 ATP synthase, which we'll talk a lot more about in the next lecture, they generate rotation in this little motor. And what that does is that torque, that rotational energy, is used to do oxidative phosphorylation. Here, ADP and inorganic phosphate in solution, not coming from a substrate, are coupled together into ATP. So this is a very different way to synthesize ATP than the substrate level phosphorylation. Yeah, I think I've done quite enough damage for one day. I'll see you guys on Wednesday.